The overall goal of the following experiment is to alter the expression of a gene and then to measure its impact on somatic hypermutation. This is achieved by transfecting BOSC23 cells to produce viral particles containing either an overexpression or an shRNA-containing construct. As a second step, Ramos cells are infected with the viruses, resulting in either knockdown or overexpression of the gene of interest. Next, single-cell clones are grown for several weeks in order to allow the cells to accumulate mutations. Finally, the effect of the altered gene expression can be determined based on analysis of IgM loss and by sequencing of the Ig genes.
This method can help identify novel factors involved in antibody diversification.
Begin by aliquoting 600 microliters of warm unamended DMEM into a 1.5 milliliter microcentrifuge tube. Then, being careful to pipette directly into the media, dilute 30 microliters of Fugene 6 into the tube. Vortex the solution for less than a second and then incubate the tube at room temperature for 5 minutes. Next, for a lentiviral infection such as this one, add 6 micrograms of shRNA construct and 1.5 micrograms each of PVSVG, PMDLG-PRRE, and PRSV-Rev to the DMEM Fugene 6 mix. Vortex the solution briefly, and then incubate the tube at room temperature again, this time for 25 minutes. Finally, transfect a BOSC23 cell culture at 50 to 60% confluency with the DNA-DMEM Fugene 6 mix and incubate the cells at 37 degrees Celsius and 5% carbon dioxide for 24 hours.